Hello everyone. So this is the week 2 question answer session of the NPTEL course Fundamentals of Protein Chemistry. Course coordinator is Professor Dasgupta from the Department of Chemistry, IIT Kharagpur, and I am Beas Mukherjee, a research scholar from Dr. Shomode's lab in the Department of Bioscience and Biotechnology, IIT Kharagpur. So today we will talk about proteins and we will solve question and answers. So these question answers will be based on whatever you have learned in the previous week. So this full week, whatever you have learned in the NPTEL course, based on that, we are going to discuss these question answers. So if you are having any doubts, please reach out. So you can type in the chat box or you can unmute yourself and you can ask the question. Okay. All right. So let the others join. For today's session, I'll give you a brief overview. So in the last class, which was our first doubt clearing session, we talked about amino acids. And we talked about the stereochemistry of amino acids. We talked about peptide bonds, torsion angles. We talked about a lot of different things. So you can go back to that. It is available on YouTube and it is the PPTs also. It has been shared with you. So today we are going to talk about proteins. So these amino acids join together and form proteins, right? This is what we discussed in the last class. So let's begin with today's class. Now you tell me, you stop me in between if you're not able to see the slides or any other doubt that you have. If I'm not able to answer today, I'll get back to you the next day. Okay, but please make sure that you're able to understand whatever I'm telling. So we will do a little bit of theory and then we'll go into the corresponding questions, and then once again a little bit of theory, and this is how we'll go so that you don't get bored. Okay, so this is going to be around 1.5 hours session. Okay, and in case you miss out, in case you're not able to continue with the session, something urgent comes up, don't worry, this entire video gets recorded, so it gets uploaded on YouTube and the session link will be provided to you by NPTEL team, plus the PPT will be given to you. Okay, so all the questions that are there, all the website links, the books that I have used here, referred to, and all the website links and everything will be provided to you. Okay, right, so let's begin. So today we are going to talk about proteins. Okay. So you tell me if you're not able to see. Okay. So what are proteins? Proteins are complex biological molecules that perform life processes in our body, and they're made up of long polypeptide chains of amino acids joined by peptide bonds. Okay. Now proteins are the second most abundant substance in our body after water. Proteins perform a vast different types of functions, and you of course know them. All the hormones in our body, most of them are proteins. Then we go to the enzymes. Enzymes are all proteins. Then the tissue, they are the cell, basic components of the cell, they are made up of proteins, right? So DNA replication, response to stimuli, catalyzation of metabolic reactions, providing structure to the cells and organisms, transport, everything is done by proteins. Okay, so in the next slide we see that proteins are very, very important in our body. This is something that we all agree with. This is something that we know. So the entire immune system, a large part of it is dependent upon proteins. Okay, next. Right. So now you might have already heard about this. This is the central dogma of molecular biology. Okay, you tell me if you're not able to see any of the slides. Okay, because sometimes what happens is I will keep changing, but then you will not be able to see. Because of network issues, this happens a lot of times. So if you're not able to see, please point out. Okay, so central dogma of molecular biology tells us how the genetic information gets transformed from DNA to RNA and from RNA to protein. Now, there is also reverse transcription. So, the genetic information can pass on from RNA to DNA. That happens in case of some viruses, right? Retroviruses. So, we will learn about all of these later on in the upcoming classes. But this is the basic. So, DNA replication happens. So, it is self-replicating. RNA information can pass on to RNA, and So according to the central dogma of molecular biology, which is proposed by Crick in 1958, genetic information flows from DNA to RNA to make a functional protein product. Now proteins differ from one another based on the sequence of amino acids, which is dictated by the nucleic acid sequence in the genes, right? So the code, the genetic code, the codons that correspond to a specific amino acid, and based on the codon that the tRNA is reading, a specific amino acid is being incorporated by the tRNA during the translation process, and this is how the entire polypeptide chain is made. That is how proteins are created, right? So this results in, and based on this sequence of amino acid, there are going to be different kinds of interactions within the protein, because of which folding happens and the 3D structure of the protein and its biological activity is obtained. So the whole process is dependent upon the sequence of the protein, which in turn is dependent upon the genetic makeup, right? Okay. So next, understanding the peptide bond. Now this is something that you have already discussed. We have already discussed in the previous class how peptide bond is getting formed when two amino acids come together and join. Okay. Plus the peptide bond is having a partial double bond character, and that is why, due to this resonance between different structures, so this carboxyl group and the NH group, so this is how resonance happens, as you can see in this slide, right? We discussed this in the previous class. So carbon and oxygen atoms involved in the bond share electrons in a way that creates a partial double bond and making the bond more rigid. So peptide bond is relatively shorter compared to a normal C-N bond, and it is also more rigid and prevents free rotation. That this rigidity contributes to the overall three-dimensional structure of proteins. Okay, now we move on. So how are proteins formed? So proteins are formed by first formation of the amino acids joined together to form the primary structure. This primary structure now folds upon itself through intramolecular interactions, that is seen in case of alpha helices, and intermolecular interactions, which is seen in case of the beta pleated sheets, which can be parallel and antiparallel. And then there are, so these are basically hydrogen bonding interactions. Next, there comes in other interactions such as hydrophobic interactions, disulfide linkages. There comes in like electrostatic interactions, salt bridges, etc. All of that is seen in tertiary structure. Next, we go to quaternary structure. So what happens is, sometimes the protein molecule yes, I hope you can see. If you're not able to follow, please let me know. Okay. So sometimes the tertiary structure is biologically active and sometimes it is not. For example, in case of hemoglobin, 4 different monomers come together and join and form the functionally active quaternary structure, right? So this is something that you have to understand. This is simple, I'm sure you all know about it, right? So now here in this slide, whatever I said just now, that has been explained better. So primary structure, the sequence of the amino acid in a protein is called its primary structure. It gives a unique identity to the protein. Secondary structure are alpha helix and beta pleated sheet, commonly the secondary structures in proteins, and secondary structure refers to the local folding patterns within a polypeptide chain. These structures are stabilized by intermolecular in case of beta sheets and intramolecular in case of alpha helix hydrogen bonding. And then tertiary structure is formed by folding of the secondary structure into a definite geometric shape, and it usually has a polypeptide chain. This polypeptide chain usually has a backbone with one or more secondary structures. So the tertiary structure is determined by the interaction and bonding of the amino acid side chain. So we will look into the details of these intermolecular interactions in the upcoming slides. Okay, so quaternary structure, I'm sure you've already understood. I gave you the example of hemoglobin, right? So more than one subunits come together to form the quaternary structure. Next, what happens in case of an alpha helix? So see this image, it's a very interesting image and try to understand this. So the alpha helix is the most common secondary structure in proteins. It is made up of 3.6 residues per turn. So what happens is, this red atom, this is actually oxygen. and the black atom that you're seeing marked as 2, this is carbon. So this is the carboxyl group, and this 3 is actually nitrogen, and the one below it that is in white, that is actually hydrogen. So this CONH, it is actually the peptide bond. CONH is the peptide bond, right? And this oxygen of one amino acid interacts with the third amino acid. It is interacting with this NH group of the third amino acid. Sorry, it is interacting with the NH group of the fourth amino acid, and that is why it is forming this alpha helix. So it has 3.6 residues per turn, right? When it is interacting with just the next amino acid, that is known as a 2.27 ribbon. If it is interacting with the third amino acid, it is called the 310 helix. So these are different types of structures that can be formed other than alpha helices, right? And then what is a pi helix? So pi helix means that it is interacting with the fifth residue. Fifth residue, right? So intramolecular hydrogen bonds between the carbonyl oxygen of one residue and the amide hydrogen of the fourth residue down the sequence, that is alpha helix. Now 2.27 ribbon and 310 ribbons are described by the notation NM, where N is the number of residues per helical turn and M is the number of atoms that come within it. So if you count starting from this 1, if you count to let's say 7 or 10 or 13, you see that there are 7, 10 and 13. That is why it is 2.27, 3, 10, right? This is how it goes. So now see, including hydrogen in the ring that is closed by a hydrogen bond. With this notation, an alpha helix is 3.6, 13 helix, right? Now, coming to an alpha helix, some amino acids are naturally capable of forming alpha helices very easily. Some amino acids are not very good with forming alpha helices. For example, glycine. Now, glycine does not have a side chain, right? So it is very flexible. Plus, if we look at proline, proline actually bends the helices, right? It forms kinks in helices, and that is why it is not a very good helix former. So some of the residues are very good helix forming residues. For example, leucine, alanine, and then methionine. So it's mentioned here, right? Lysine. All of these have high alpha helical propensity. So they are very good helix formers. And how have people come across this? So this alpha helical propensity, this can be tested using CD spectroscopy, right? So they are compared to glycine and proline, which are often called helix breakers due to their side chain properties. Okay. Now we move on to the properties of the beta sheet. Now this is a very interesting part, and you will get a lot of questions from here. You can expect questions from here. So one of the major questions is that in nature, are parallel beta sheets stronger or are antiparallel beta sheets stronger, and why so, right? So see, in antiparallel beta sheet, the hydrogen bond, it is denoted with this dashed arrows. These hydrogen bonds, these are actually just 90 degrees to the beta sheet, right? But for parallel, it is actually zigzag, right? So in parallel beta sheet, the beta strands are unidirectional. However, the intermolecular hydrogen bonds are not perpendicular to the axis. They are all zigzag. In the antiparallel beta sheet, the consecutive beta strands run in opposite directions. So for example, if this is going from N to C, the next strand will go from C to N, right? This is how it goes. The intermolecular hydrogen bonds are exactly perpendicular to the axis. In the mixed sheets, now there can be mixed beta sheet where parallel and antiparallel are there together. So parallel sheets are less twisted than the antiparallel sheet and are always buried. In contrast, antiparallel sheets can withstand greater distortion, twisting and beta bulges, and greater exposure to solvent. This is why the antiparallel beta sheets are more stable compared to the parallel beta sheets, which is consistent with both hydrogen bond geometry and the fact that smaller parallel sheets occur in nature. That means that because of the length of the hydrogen bonds, you know that bond length is inversely proportional to bond stability. right? So here, when they are exactly perpendicular, the bond length is relatively small, but when they are zigzag, the bond length is more. So if the bond length is small, then the bond strength will be higher, right? Because of which even fewer number of antiparallel beta sheets can exist independently in nature. But if it is a parallel beta sheet, there will be, I think, minimum 5 or so parallel beta sheets have to be present to be there in nature as a secondary structure in a protein. Okay, so this is one of the very interesting things that you can keep in mind. Right, so we move on. Now, interactions in a protein. So in a protein, there can be hydrogen bonding interactions, there can be hydrophobic interactions, that is clustering of the hydrophobic groups away from water, and van der Waals interactions. There can be disulfide linkages between two cysteine residues. There can be ionic bonds. Okay. Now, based on whatever we have studied until now, what do you conclude from the structure of this protein? So this is a protein, it's an RNA binding protein, and we work with this in our laboratory. Okay, so based on whatever you have learned until now, what do you conclude about the structure of the protein? What can you see? So how many alpha helices and how many beta sheets are there, and what kind of beta sheets are there in this protein? You can type in the chat box, I'll be able to see. Right, you can just give it a try. Okay, so this protein is actually made up of 2 alpha helices and 5 antiparallel beta sheets, as you can see from the figure, and its biologically active state is actually the tertiary structure. So we are not seeing this as a monomer, we are not seeing this as a dimer or trimer, and this is biologically active. It's PDB, like its structure is there uploaded on PDB, protein data bank. Okay, right. So now we move on. So torsion angles in proteins, I will not go into this anymore because we have already discussed this in the previous class. Right, so I will not be going into this. So torsion angles in proteins and the entire Ramachandran plot was discussed in the last class. Now we go for the assignment questions. Okay, this is a simple question at first. So try to solve this question. Primary structure of a polypeptide is formed by what kind of reaction? So this also I'm sure you can answer based on what we discussed in the previous class and what we have learned until now. Okay, whether it's a condensation reaction, whether it's a substitution, elimination or hydrolysis. A. Okay, very good. Yes, that's right. It's a condensation reaction because in condensation reaction, two molecules, they are joining together and leaving out this water moiety. Right, this is what is happening. Okay. Now there is another question. This is also a simple. So you tell me something, when I'm changing the slides, are you people able to see or are you just seeing blank screen? Because then I will have to change it. Okay, you try to answer this question. Which of the following bonds within a peptide backbone do not show free rotation? So we discussed this today as well as last day. This should be simple, right? So we know about phi, we know about psi. So in phi it is NC. Is it NC? Is it NC alpha? Let's see if you remember. One of the bonds is, yeah, okay, so B. B is CO and NC. Let's see. Let's see the right answer. Okay, so the right answer is actually NC. NC is this peptide bond and C alpha C. One second. C alpha C is psi. Due to conjugation, NC is definitely the right answer, but then I will actually have to check. It can, I'm not sure if this is C alpha C because C alpha C is psi. So psi actually has free rotation. Okay, I'll check this answer once again. Okay, Right, so we move on to the next question. Okay, so which secondary structure has a strong unidirectional dipole moment? Now this is a very interesting question because I'll tell you something very interesting once you answer this. Which secondary structure has a strong unidirectional dipole moment? So dipole moment means that one end of it has a particular charge, the other end of it has a different charge. Right, so alpha helix, parallel beta sheet, antiparallel beta sheet, and beta turns. What are turns, by the way? Turns are actually what comes in between these secondary structure. So for example, this beta sheet is going this way, here is a helix. So just in between this tiny stretch of protein, this is a turn. Okay, it's kind of like a linker. So this is a beta turn. A. Okay, yes, that is correct. Yes, alpha helix is the right answer. But why is that so? For that, I will explain this thing to you. It's a very interesting concept. Try to see it this way. Okay, you have studied in school about magnets, right? So magnet is having this, even if it is like very tiny, it is going to have this north pole and south pole. Correct. Now amino acids, they actually behave like tiny magnets. Amino acid is having COO minus group and NH3 plus group, right? So what happens is all the amino acids are in a helix. So all the, okay, 1 second, someone is trying to join. Yeah, I'm facing some network issues. Okay, 1 second. Right. So what happens is all of these tiny amino acids, they behave like tiny dipoles. Here they are having positive and negative charge, right? And all of these amino acids, they are placed in such a way in a helix that all the dipole moments, they point in the same direction, because of which the entire helix behaves like a big dipole. This is called helix macrodipole. Okay, and this entire helix has a dipolar moment. So it has a more positive N terminus and a more negative C terminus. So see, this dipole moment of the constituent amino acid give a net macrodipole to the alpha helix. And 1 second, someone wants to join the call. I'll just let this person in. Okay. I hope they have been able to join. Okay. All right. Let's continue. So, and therefore the N terminal of the helix is more positively charged and the C terminal is more negatively charged. Right. Okay. So now we move on to the next question. Okay. So see here, this has been explained better. So this amino group is basic and has a lone pair of electrons on the nitrogen, making it polar. A carboxylic group is acidic and has a polar CO bond and a polar OH bond. The molecular geometry. So the carboxylic group and amino group are on the opposite ends of the molecule, creating a dipole. This is what I explained to you, right? And electronegativity differences. So the atoms involved, such as N, O, and H, have different electronegativities. This difference in electronegativities between the atoms leads to uneven distribution of electron density, resulting in partial positive and negative charges, which contributes to the dipole moment. Right? So you can read about this, and there are many interesting things about a helix. So when see, I work in a protein engineering laboratory, right? So when we are trying to stabilize proteins, we need to keep all of these different factors in mind. So based on all of these, we are introducing mutations in the helix and we are trying to stabilize the helix. So these are called helix stabilizing mutations, right? So these are different things. So this box is there with a lot of information. You can go through it. So what is a helix cap? Helix cap is actually between the first and the, yeah. So some residues, when they are present in the beginning and end of a helix, they form this double reciprocal hydrogen bond. For example, the side chain group is interacting with the main chain of one amino acid, and again main chain of this particular amino acid is interacting with the side chain of this amino acid, and this is how it forms a very stable structure at the two ends. This is called helix capping. Some residues are known to be very good helix caps, helix cap formers. Okay, so they are present in that way. So you can go through this tiny box that is given in this slide and have a better understanding. Okay, but we'll move forward with the questions because there are a lot of questions and there are a lot of concepts that we need to touch upon today. Okay, so this is a simple question based on your understanding. Okay, let's see if you can answer this. The molecular weight of a polypeptide can be very precisely determined by, okay, the key word is precisely. Now let's see if you can answer. So agarose electrophoresis, SDS-PAGE, MALDI-MS, and elevation in boiling point. I don't know where elevation in boiling point is coming from, but let's see if you can think of something. Okay, so the molecular weight of a polypeptide. Yeah, okay, C. Yes, that is very correct. So a lot of you will actually tell me that why even SDS-PAGE can be used for determining molecular weight, right? But then the term precisely tells you that yes, only mass spectrometry is the only way using which you can very precisely know the molecular weight of a polypeptide. You can know the entire amino acid sequence of that polypeptide, right? But from SDS-PAGE, from size exclusion chromatography, you can know the mass, but then not the exact definite mass, right? So it can give an idea about the molecular mass, but it cannot determine the absolute molecular mass. Okay, now how does SDS-PAGE work? I am sure a lot of you have already done this in your practicals. A lot of you already have some idea, but some things, even if we are doing it every day, we keep on forgetting, right? So this is just to give you an overview of the principle. The principle of SDS-PAGE is something that you're going to need for all exams because this is a very commonly used technique, but it's very important because there are certain very tiny things that you need to keep in mind about SDS-PAGE. So see, the protein is normally folded, right? Proteins can have positive charges, negative charges based on the pH and based on the amino acid composition. Now, if you let that into a gel and if there is, let's say, current flowing, what will happen? Some protein will move upwards, some protein will move this way. Based on the charge on the of the protein, it will move, right? But then we will not be able to figure out anything from that gel. That is why what we do is we give all the proteins negative charge. Now what happens? The proteins will migrate on the gel. They will move towards the positively charged electrode, and based on the mass difference, we will be able to say which protein is heavier and which protein is lighter. And then we run a ladder of known molecular weights, and this is how we can know about the mass of a particular protein. Right. So what happens is we introduce beta mercaptoethanol. So beta mercaptoethanol breaks down all the disulfide linkages, and then we heat the protein. We denature the entire protein. Then what happens? Then we put in a lot of SDS. SDS is sodium dodecyl sulfate, which is a detergent. It's an anionic detergent, and this gives negative charge to the protein. Okay, so and what happens? Heating actually denatures the protein. That means its natural fold is gone. Now it is a linear chain, and then this SDS molecule comes and sits on it, gives it uniform negative charge, and then when you run it, what happens is it migrates only on the basis of difference in molecular weight. So this is something that you have to remember. Now in the gel also there is something very interesting going on. Okay, now one of the questions is how many, let's say you're giving this SDS. So how many SDS molecules are going to bind to each amino acid, or whether like that is a very interesting question, and it is possibly there in this slide, or maybe like it's a very common questions that they can ask for exam. So just for your knowledge, you have to find out the answer and remember. Okay, if it's there in the slide, well and good. If not, try to find out the answer. Okay, so see what happens is it is made up of a two type of gels. Okay, so it's called a discontinuous gel system. Why discontinuous? Because there are two different types of gels. It's actually a vertical process. Okay, agarose gel is horizontal, but this gel, polyacrylamide gel, it's actually vertical. So the below gel, it's made up of, it's called resolving gel, and the difference is that it has a pH of around 8.8. Okay, pH of tris chloride. In the stacking gel has a pH of around 6.8. Okay, now in the stacking gel, what happens? It has larger pore size, and the separating gel has smaller pore size. So stacking is important because as you load the protein, as you put the protein into the well, we don't want it to immediately start running, right? Because there can be differences in the way it initially migrates through the gel. That is why we want all the protein to come to a particular level and then start the migration process. So for that, stacking gel is there. Now there is a very interesting principle behind this. So see, in the stacking gel, in the buffer, there is a lot of chloride ion, there is a lot of glycine, and there is a lot of SDS. Okay, in the stacking gel, the charge of glycine is such that the glycine runs above the protein. Your protein is like a sandwich in between, and then the chloride ions, obviously it is smaller molecular weight compared to the protein, so it will run faster than the protein. Then what happens? As it migrates, it goes into the dissolving gel with pH 8.8. Here what happens? The charge on the glycine changes, right? Charge on the glycine changes, and now the protein runs behind, glycine runs faster, and chlorine is the fastest. Now how does that happen? Let us look at the next slide. So see, here you'll get to know. At the stacking gel, the counter ion or glycine remains neutral. It's a Zwitter ion. Zwitter ion means it is having equal number of positive and negative charges. And the chloride ion is the principal current carrier, and it migrates towards the anode, which is the positively charged electron. The negatively charged SDS protein complex are stacked as a thin band between glycine and chloride ions. In the resolving gel, glycine forms glycinate ion. So it becomes negatively charged because the pH of glycine is around 7, right? It's a neutral amino acid. Its pi is around 7. That is why what happens is it becomes negative once that is crossed, and we see that in the resolving gel, the glycine forms glycinate ions and follows the chloride ions. So the SDS, sorry, one second. Yeah. And the SDS protein molecules are well resolved by the molecular sieving effect of the pores based on the size difference, right? So there, just on top, it is given how glycinium cation is there at a very low pH, and then as pH becomes neutral, it becomes Zwitter ion, and then at a higher pH, it is becoming negatively charged, right? So at 6.8, it is a Zwitter ion, and then it becomes negatively charged. That is why there is this difference in migration of glycine, because of which you are getting this stacking effect. Now we'll move on. Okay, so how does mass spectrometry work? So mass spectrometry is a very interesting question for examiners, and it's a very interesting concept. Here it is given as in a very compact way, but then you have to read about it properly from books. So I have referred to books. Okay, one second. In this particular slide, you can actually refer to this virtual labs link. So this is something that we all have developed, and what happens is here you can actually perform the experiment virtually. Okay, this is for you to understand SDS-PAGE nicely. So me and my colleagues have developed this, and you can actually go through this. It will give you a very good understanding of the entire concept of SDS-PAGE. Okay, besides SDS-PAGE, there are also many other experiments. You can go through those. Okay, so now what happens in a mass spectrometry? So in mass spectrometry, okay, so there's a difference between spectrometry and spectroscopy. Spectroscopy means you're analyzing the interaction of matter with light. We had studied about spectroscopy in the previous class. But spectrometry means that you are measuring something. Okay, so mass spectrometry is an analytical tool in which the samples are ionized into charged molecules, and then based on the mass by charge ratio, they are measured. So in MALDI-TOF is a type of mass analyzer. So TOF means time of flight. It's a mass spectroscopy. The ion source is matrix assisted laser desorption ionization. right? So even the full form comes as questions in exams. So it's important that you remember this. And the mass analyzer is a time of flight analyzer. So what happens here is MALDI is a technique that involves laser. So there is a matrix on which your sample is there, and there are also resin particles on the matrix, and the laser falls on this, and because of that, this initially it was a liquid. This liquid actually gets ionized and it turns into gaseous particles. So to make the analyte molecules into gas phase without fragmenting or decomposing them, right? Next, what happens is, see, so initially there is this target plate onto which the analyte is present, and matrix particles are there. Laser beam falls on it, and then the analyte gets separated. It forms a gaseous particle, and on this gaseous particle, the matrix actually coats it, and the matrix is having some charge. Because of this, it is giving it some charge. Right, so analyte is embedded in a matrix compound solid support called target, made up of a conducting metal. After a brief laser pulse, UV or IR, the irradiated spot is rapidly heated and becomes vibrationally excited. The matrix molecules are energetically ablated from the surface of the sample, absorb the laser energy, and carry the analyte molecules into the gas phase as well. During the ablation process, the analyte molecules are usually ionized by being protonated or deprotonated with the nearby matrix molecules. The most common MALDI ionization format is for analyte molecules to carry a single positive charge. So normally these analyte molecules will carry single positive charge, and they will pass through this. Once they are getting vaporized, they are getting into gaseous phase, they will pass through this time of flight detector, and this detector, it is actually having these so based on the migration of charged molecules in the magnetic field, the time of flight analyzer is going to detect its movement, and based on the mass by charge ratio, it is going to characterize it. So all the ions are given the same initial kinetic energy. So the ions of the same mass by charge should reach the detector at the same time. Right. So let's continue. There was some network issue. I kind of got disconnected. Right. So however, in practice, the pulse is not felt by all the ions to the same intensity. And so not all the ions of the same mass by charge values reach the ideal velocities. To correct this problem, a reflection is used. It consists of a series of ring electrodes with high voltage which can repulse the ions back along the flight tube, usually at a slightly displaced angle. So this can sound too difficult for you at first, but then once you read about it, it will get easier, and then you will be able to understand this properly. Okay. Now, see what happens is faster ions carrying more kinetic energy will travel a longer path than the slower ones, and thus spend more time within the reflectron than the slower ions carrying less energy. Then what happens in that way? The detector receives ion of the same mass at about the same time. So thereby the design of time of flight mass analyzer has increased the resolution. So this is a very useful technique, mass spectrometry. You should read about it, and it's very important for your exam point of view as well. So your whatever exams that you're appearing for, be it GATE, be it CSIR NET, be it DBT, these are questions that you can expect. Okay, now there is something very interesting, and I have included this for the sake of an MCQ that we will come across. So all the MCQs that I am going to discuss here, these are actually taken from previous year's NPTEL questions. Besides that, we have our, one second, facing this network issue. Okay, I'm so sorry about that. There was some network issue here. Okay, so there was some network issue here. I hope we are connected still. Okay, so let's look at contact maps. Now, contact maps is something that is a 2D representation. Now, this is important for one of the MCQs that was previously asked by NPTEL, and all the MCQs that we will be discussing here, these are either previous year's questions or they are questions that are taken from different sources, which can be a different NPTEL course that I took. Okay, it can be other questions, it can be new questions as well. So it's very important that you pay attention and practice these questions, okay, especially the concepts, because it's very diverse, right? The syllabus is, it's huge, but then what to study and what not to, that you get an idea from the questions. That is why it's very important to go through as many questions as possible. Questions give you an idea about what you need to look into and what not. Okay, so contact map is actually a distance matrix. It is a 2D representation of the distances between the pairs of atoms, usually C alpha atoms in a proteins and other macromolecules. It is used for the understanding of the three-dimensional structures and secondary structure elements in a protein. So it is a square matrix where each element I and J represents the distance between the ith and the jth residue. Okay, so one of the axis is this and the other axis is this, and the diagonal that you're seeing is the same element. So if the distance is between a certain threshold, usually indicating a contact, the element is often marked by a dot or a color. So all these small black dots that you see, these are interactions between different residues. Okay, the threshold is typically set to identify interactions that are relevant to the protein structures such as hydrogen bonds, hydrophobic contacts, and van der Waals interactions. The line in the diagonal, this is for the own interactions with itself. That is, so we look at this bit. Right, so see here, everything is explained properly. The diagonal line, the main diagonal from the top left to the bottom right, represents the distance of the residues to themselves. So distance is 0. It is always marked, right? Off diagonal elements. So off diagonal elements represent the distance between different residues. If this is the diagonal and this is an element, this is an off diagonal element. A secondary structural element. So alpha helices appear as continuous blocks along the diagonal. This is because residues in an alpha helix are in close proximity with their neighbors. So for example, if we look at this figure once again, so see, the alpha helix is there as continuous blocks along the diagonal, right? So this is then an alpha helix, right? It is along the diagonal. Next, beta sheets, they appear as off diagonal patterns. They show up as lines parallel to the main diagonal. Okay, sorry. So this is then a beta sheet, right? So this is a parallel beta sheet. An antiparallel beta sheet, they are as lines perpendicular to the diagonal. So interpreting the contact map. So how do you interpret the contact map? So the clusters of the patterns, dense clusters or points indicate regions where many residues are in close proximity, suggesting a tightly packed secondary structure. And sparse regions are regions that indicate where the residues are further apart, possibly indicating loops or structured regions. Now let's try to solve this question. Okay, right. So there is this Z, there is this X, and there is this Y. Now, based on your understanding until now, which one do you think is which? Okay. One is along the diagonal, one is perpendicular to the diagonal, and the other one is parallel to the diagonal. Right? So, try to identify X, Y, and Z. You can answer in the chat box. One is along the diagonal. Which one is that? Alpha helix is along the diagonal. X is okay, and your parallel beta sheet is parallel to the diagonal, and anti-parallel beta sheet is perpendicular to the diagonal. So which one do you think is which? You should be able to answer. It's not very difficult. Try. Okay. So see, this is the answer. So parallel to the diagonal axis and adjacent to the same is due to alpha helix, which is marked as Y. That is your alpha helix. So alpha helix is Y, right? Parallel to the diagonal is due to parallel beta sheet, which is marked as Z. And perpendicular to the diagonal axis is due to anti-parallel beta sheet marked as Y. Okay. I hope you've understood this. Right. Now we move on to something that you already have enough idea about. I'll not explain this in details. You already know it. Right. So fibrous and globular proteins, all the proteins that are soluble and they are spherical in shape, they are globular proteins. All the proteins that are not very soluble and they are majorly made up of beta sheets, they are fibrous proteins. For example, the hair, keratin is a fibrous protein, right? So they help in the fibrous proteins are long and thread-like and they are insoluble in water. Example, keratin, collagen, and elastin, fibrin, etc. They provide a structural framework. Globular proteins, they are soluble in water due to their hydrophobic R groups are located in the center. That is why the outer side is hydrophilic, right? Example, hemoglobin, myoglobin, insulin, etc. Right? So they are very important for the different metabolic and chemical processes in our body. Now, fibrous proteins, collagen and keratin. Let's have a look. This might sound very repetitive, but this is something which from where you get questions very often. Okay, so it's very important that you go through all of these properly. So see, keratin, these are monomers. The keratin monomers are actually left-handed supercoiled. So they are left-handed supercoiled to form very stable superhelical or a coiled structure. Along the length of the filament, there are hydrophobic interactions between the non-polar amino acids and thus maintain this coiled structure. Okay, keratin displays disulfide cross-linking. So the curly hair, right? A lot of people have curly hair. We are Indians. A lot of us have curly hair, right? And what we do is we use heat to actually straighten the hair. So this heat treatment is actually breaking your the interactions in the keratin, and that is how you're getting this what do you say, straight hair. And that is why heat treatment is a very temporary thing, right? Because heat denaturation, it can be reversible, but then chemical treatment and all, that is not reversible anymore. So that is something very interesting. You can look into that. And then collagen. So the left-handed helices are twisted together into a right-handed triple helix or super helix, and in a cooperative quaternary structure stabilized by many hydrogen bonds in a triple collagen triple hydrogen bond form between separate chains. Okay, so collagen is made up of 3 left-handed helices twisted together into a right-handed triple helix. That is very interesting. Now the combination of these amino acids, now this is the place from where you get question. What is this tripeptide repeat in collagen? What are the most common amino acids in keratin? So these are the questions that you can expect. Okay, so glycine, proline, hydroxyproline. This is the one that is there in repeats, and this prefers a conformation. So hydroxyproline always comes from the surface of the structure, and there are different post-translational modifications such as hydroxylysine, etc., and crosslinks in collagen, which includes formation of hydroxyproline and hydroxylysine. Right, so there will be a lot of questions from this collagen and keratin. So please make sure that you remember all this. Okay, now we have a small question. Try to answer this. Which of the amino acids are most abundant in collagen? Proline, aspartic acid, glycine, glutamic acid. So you have to select 2. Try to answer. You can answer in the chat box if you want. Okay, this should be simple. We just talked about this. Okay. Let's look at the answer. So 1 and 3 is the answer because we have glycine, proline and hydroxyproline, right? So 1 and 3, these 2 residues are there. So this is how you can see the diagram that this glycine, hydroxyproline and proline, these are all present, and these form this 3 individual polypeptides which twist together to form this structure. So this is what you can see here, right? And these repeats form these alpha chains, and these alpha chains come together to form the triple helices, and triple helices form the collagen fibrils, and the fibrils form the fibers. This is the process. Okay. Now, there is one more question. Try to answer this question. The interacting surface of a membrane protein with the membrane is? Which part of a membrane protein interacts with the membrane? So first, for this you have to think about the structure of a membrane, right? So in the membrane, you know how membrane proteins are embedded. So membrane is made up of hydrophilic region, hydrophobic region, right? So the lipid molecules are there. Inside of the membrane is hydrophobic and the outside of the membrane is hydrophilic. And the protein, if it is the interacting surface of the membrane protein. So membrane proteins can be of different types. There can be transmembrane proteins, there can be surface proteins, there can be different types of proteins. So which one do you think would be the answer here? Okay, we'll look at the answer. So hydrophobic, because see, this is the bilayer, and in the bilayer you can see that these protein molecules are actually embedded deep inside, and the transmembrane region is hydrophobic in nature, and this is the one that is interacting with the hydrophobic tail of the lipid of the membrane. Right, so middle part of the membrane is hydrophobic, hence to interact with the membrane, the protein has to be hydrophobic. Okay, this is something that you know from the fluid mosaic model, which you studied in school, possibly, right? Okay, let's move on. And this is another question. The factors responsible for adaptation of a particular protein conformation in aqueous solution. So we studied about globular proteins, and there we learned about something interesting, right? That all the hydrophobic regions, hydrophobic residues, they are mostly packed within the core of the protein. Hydrophilic residues are on the outside. Okay, these are the ones that are constantly interacting with water, and that is how the protein becomes soluble. So here they are asking you what are the different factors that are important to give a protein a particular conformation in an aqueous solution. what is a conformation? So see, the thing about proteins is very interesting. We see the structure of proteins as something very rigid, right? But in reality, proteins are not rigid at all. They are constantly moving like this. So if you look at some kind of molecular dynamics simulation video or any kind of YouTube video in which they show motions dynamics of proteins, you will see that these protein molecules, constantly there is some kind of interaction between the molecules, then there is some kind of interaction with the water around it. So some bonds are breaking, some bonds are getting formed, and constantly, let's say this is how the protein initially looks like, and this constantly it keeps on changing, right? So these are the small conformations that the protein attains. So what is the reason for that? So this conformational dynamics can be studied very nicely using NMR spectroscopy. We'll discuss about this later on in a particular session, I think. It will come up. So see, formation of the maximum number of hydrophilic interactions, minimization of entropy by the formation of a water solvent shell around the protein, placement of hydrophobic amino acids within the interior of the protein, placement of polar amino acid residues around the exterior of the protein, formation of the maximum number of hydrogen bonds. Which one do you think would be the right answer? So these are the kind of questions that you can expect in the exams. Some questions will be straightforward, but some questions will be like this. They'll be a little twisted, and the more you practice, better idea you'll have. That is why it's very important that you practice the previous year's questions. Okay, for every exam. Previous year's questions are something that guide you as to how questions can be. Right. So, let's look at the right answer. So, okay, all 5 options are correct. Is that so? Okay, let's see. So, let's see this unfolded polypeptide chain. And what happens is, in protein folding, the hydrophobic regions are majorly packed inside, and this hydrophobic effect minimizes the hydration layer and facilitate weak interaction between the amino acid residues, right? So the third one, so the placement of all the hydrophobic residues within the interior of proteins, this is a very important aspect, and placement of the polar amino acid residues on the exterior of the protein, this is also a very important aspect. So, as only 2 options are, you can choose only 2 options, right? In for all of these different options that you have, A, B, C, D, only 2 what do I say, statements can be chosen. So the most appropriate would be 3 and 4. That is why the right answer would be C. Okay. Now we move on. Right. So now see, this is an interesting question, and these are the kind of questions that you can expect. Okay, in NPTEL exam and also in your other exams, right? All the competitive exams. So see, an hypothetical helix has a 5.6 residue per turn with a 5.2 angstrom pitch. So what is pitch? Pitch is actually, let's say this is the turn, the distance of the distance between one turn and another turn, that is the pitch, right? You know pitch possibly from this DNA double helix structure of Watson and Crick, right? There also we had pitch. Here also pitch comes in. Okay, so what is the rotation angle per residue of that helix? So how do you find that out? So this is something very interesting. So I'll tell you how to solve it. For example, if I give you an example, see, there is something called a helical wheel. So what happens there is, let's say that a particular amino acid, let's say that this alpha helix, it is made up of 3.6 residues per turn, right? And so in this entire 360 degree, if you divide that with 3.6, you get that each residue is separated by 100 degrees. So see, the first option is 100 because for alpha helix it is 100. But for this one, what would be the Right answer. That is something that you'll have to find out. Okay, for the next question, dihedral angles phi and psi for beta sheet respectively are what? So this you can get from the Ramachandran plot. That is why I told you that Ramachandran plot is very important. Okay, so B is the answer for the first one or the second one? First one. Okay, okay, great. Let's look at the right answer. Yes, that is correct. So see, rotation per residue is 360° divided by number of residues. So number of residues is 5.6. Right, so that is 360° divided by 5.6, which is 64.3 degrees. Right, so that is how we are getting 64.3 degrees. So these are very small questions, but these can come as numerical aptitude test in GATE. Okay, so that is the key actually. Practicing more and more will give you some idea. Here you did not even have to consider this pitch, right? So that is how you get to know. Okay, so for parallel beta sheet, see, these are the different values: antiparallel, left-handed, right-handed. You have to remember this. This is the bad news. I'm sorry to give you this bad news, but yeah, you have to remember this if possible. If not the exact value, at least whether it's positive or negative, that you have to remember, because these come as questions. In every exam, you can expect at least one question from Ramachandran plot. Okay, this is one of examiner's favorite topics, so you can't miss it. Right. Now this is some difference between hemoglobin and myoglobin. I will not go into this. You can go through this by yourself. Right. So we already know some of these. So hemoglobin transports oxygen in blood and myoglobin helps in storage. Hemoglobin is made up of 4 different monomers. Myoglobin is a single monomer. So there are different things. So hemoglobin works by cooperativity. Myoglobin does not. These are the different differences that you can go through. Now this is important because see these tiny things such as Bohr's effect. That is, hemoglobin is sensitive to pH changes. So you have to read about these. There are questions about hemoglobin and myoglobin, and one has a sigmoidal curve for oxygen binding and the other one has a smooth hyperbolic curve, and the sigmoidal curve comes from this cooperativity effect. That is, initially it is low, but then it rises suddenly and then gets saturated, right? So these are things that you have to remember. Now this I have included because of this particular question, right? So which property makes hemoglobin and myoglobin different? So oxygen saturation profile at different pH, cooperative binding of oxygen, iron binding site, and large segments of alpha helix with spiral shape. So see which are the different things that influence hemoglobin and myoglobin. If you want to have a look at this once again, this slide, you can actually do that. So let's have a look at this slide once again, and from here you can check. Okay. Okay, and one more interesting thing is that hemoglobin is influenced by allosteric regulators such as carbon dioxide and pH level. Right, myoglobin is not influenced by allosteric regulation. That is why it has this hyperbolic curve, but hemoglobin has the sigmoidal curve. Right. So now try to answer this question. Okay, let's see. We can actually look at the answer. So see, it shows higher oxygen saturation at lower pH and hemoglobin shows cooperative binding. So which one would be the right answer? 2 would definitely be there and 1 would be there. So the right answer would be A, because 1 and 2 are there, right? So the right answer would be A. Now that's all for today. These are the different references. The first two are 2 books which you can refer. These are Leninger's Principles of Biochemistry and Voigt and Voigt's Biochemistry. So you can go for the 4th edition or any other edition if you want. And it's very important because these books are available online for free. You can go through these. These are very standard books that you can refer. Besides these, there are all of these other places from where I have taken these different images, the theory portion. So you can actually go through those and you can get a better idea of whatever we have discussed today. Okay, that's all for today. We will meet next week once again with a lot of new concepts and a lot of questions. So if you have any doubts, you can ask me. Otherwise, we can end the call. Okay. Thank you for your time. Thank you for your patience. Thank you so much.
Thank you, ma'am.
Thank you so much. So if you don't have any more doubts, we can end the session today. Okay, we will meet once again next week.